Hello, my dear students. Welcome to my today's online class on one more chapter of applications of biotechnology, that is about animal improvement or breeding techniques. Namely, to date, two such techniques are available. One is artificial insemination, AI, and another one is superovulation and embryo transfer, abbreviated as SOET. Sometimes SOET is also referred to as MOET, that is multiple ovulation and embryo transfer. So in this class, we are going to learn about two techniques which are meant for improvement of animal populations, that is AI and SOET. So I am Dr. Meera BK, Associate Professor and Head, belonging to Department of Zoology, Maharani Science College for Women, Palace Road, Bengaluru 1. You can see my cell phone number and mail ID on the slide. After the class, for any clarification, you are most welcome to contact. So this picture is telling you about the first technique, that is artificial insemination done through rectal palpation. I'll come to it a little later. And then the other one is the schematic representation of various steps involved in SOET or MOET. And let us begin the class now. See, the manipulation of reproduction in animals and human beings are collectively studied under the heading ART, that is Assisted Reproductive Technology. When it comes to humans, sometimes ART is referred to as MART, that is Medically Assisted Reproductive Technology. What all manipulations you do come under this heading. Ultimately, whatever you do, the main objective of ART is to increase the number of progeny of the desired species. First we shall learn artificial insemination. As the male produces millions of sperms in one ejaculate, the semen can be used to produce several offsprings at a time, theoretically speaking, and this is made possible through artificial insemination or AI. What are the various steps involved in this technique? Semen collection and storage. The semen ejaculate is collected, appropriately diluted, and examined under the microscope for the number of motile normal sperms. And this is done in a bull, please note. How the semen is collected? There are 2-3 methods: mechanical method, electrical method, right? And right now I want you to go back to your previous semester, that is Sem 3, paper 3. You have learnt in paper 3 zoology, applied zoology, about dairy, poultry and fisheries, right? In the dairy chapter, you must have studied how exactly the semen from the bull is collected. There are 2 to 3 methods. So go back to your previous semester, make a separate note of it, take it as homework. And once the semen is collected, it needs to be appropriately diluted. Again, there is a correct, neat protocol for dilution of the semen. You have to follow that protocol and dilute it. Again, you should go back to your previous semester and make a note how the semen is diluted. Once it is diluted, you have to observe under the microscope for the normal sperms, in the sense motile sperms. See, to give you an idea, 0.2 ml of bull semen contains about 10 million motile sperms. That means that many eggs can be fertilized, theoretically speaking. And this diluted semen, you can use it as fresh within a few days if you are using it, or you can always subject it to cryopreservation. What is cryopreservation? Preservation of the sperms, embryos, anything at lower temperature around minus 196 degrees Celsius in liquid nitrogen for long-term storage and transport. This technique is known as cryopreservation. You will preserve the semen at minus 196 degrees Celsius in liquid nitrogen, and once you do so, you can export it to different countries, okay, where the same can be applied, same can be made use of, okay. Then if you do so, the semen transport does not involve any quarantine measures to be followed. Why I am telling this is, suppose if you want to transport the bulls as such to a different country, so that you want to produce the progeny of high quality, you need to follow several quarantine measures as put forth by both the governments. But for sending the semen, no such quarantine measures are needed. You can just put it in a small vial, post it. That's what I meant by semen transport does not involve any quarantine measures, right, to be followed. Artificial insemination is done on standing animals through rectal palpation. How exactly is done is shown in this photograph. You can see a technical person. He has put his left hand into the rectum just to palpate that area of a bull, and then in his right hand he has a syringe containing the semen. So he is going to inject the semen right into the female genital tract. This method is known as rectal palpation. So by this method, each semen ejaculate of a bull can be used to inseminate as many as 500 cows at a time. And this semen can also be transported to different places, I have told you, countries, to be used and to produce superior animals. Ultimate purpose of any such technique is to produce quality progeny. That's all. The second step in artificial insemination is synchronization of ovulation. How it is done? What is ovulation? Ovulation is release of ovum from the Graafian follicle in an estrous cycle. By now you must be knowing that all non-primate females, mammals that is, practice a reproductive cycle known as estrous cycle, and this estrous cycle has different steps, 4 steps, and only during this estrous phase of estrous cycle the females can be inseminated, and this lasts only for a few hours in cows. Okay. So you have to do this artificial insemination in the females after ovulation, that is release of the ovum, which can be detected by behavioral estrus. Okay. When the animal is in estrus, it shows particular typical behaviors. So by studying the behavior of the cow, you can find out whether she is in estrus phase, and then you can inseminate her. But normally it is difficult to detect the estrus phase because it occurs during night and lasts only for about few hours. Now, animal breeders, they like to inseminate a large number of females simultaneously so that the management becomes easy. How to do this? All the cows may not naturally attain their estrus phase simultaneously. For that, you can go ahead with this procedure, that is synchronization of ovulation. The females are synchronized to ovulate at a set of time. You can see to it that all the cows will undergo ovulation in a particular time. Okay, you can bring about this. How? The synchronization of ovulation is brought about by exogenous administration of the hormones progesterone and prostaglandins, that is PGF2A. There are different types of prostaglandins. Here you have to make use of PGF2A, prostaglandin F2A. Progesterone, it is a female reproductive hormone. And FSH. FSH is one more hormone, follicle stimulating hormone, elaborated from anterior pituitary. Okay. Whereas progesterone is secreted from the ovary itself. But if you are planning to have synchronization of ovulation, say in 100 cows, you have to give exogenous doses of these hormones so that all the cows are brought to this estrus phase simultaneously. If you try in 100 cows, the success will be around 85%. 85% of the cows, 85 cows will come into estrus, and in all the 85 you can do this artificial insemination. Okay. So that's what I have told you. This is one technique, artificial insemination. Then another technique is superovulation or multiple ovulation and embryo transfer, abbreviated as SOET or MOET. In routine estrus cycle, normally one ovarian follicle out of 20 matures, releasing one fertile egg at a time. Please note, there will be many follicles developing in the follicular phase of estrus cycle. Around 20 will be developing. Out of 20, ultimately it is only one ovarian follicle attains maturity, and that one will release the egg. This is what normally happens in regular routine estrus cycle. And please note, whether it is estrus cycle in non-primate female mammals or menstrual cycle in primate females, both the processes are under neuroendocrine regulation, that is hormonal control, hormones from ovary, pituitary, and neurohormones. Okay. So here also, by exogenous administration of these hormones which regulate estrus cycle, you can always see to it that the cow, instead of producing one fertile egg, produces or releases more mature eggs, around 8 to 10 out of 20. Instead of one developing, attaining maturity completely, 8 to 10 will attain maturity at a time. And this procedure is known as superovulation or multiple ovulation, right? So this concept again depends on various exogenous factors and also endogenous factors like nutrition, health of the animal as endogenous factors, and environmental factors as exogenous factors. This picture gives the schematic representation of the soyet or moyet. You can see a sire of high genetic merit. This is the bull. It has so many virtues, so many positive qualities. You would like to have more progeny out of the sperms produced by this bull. Okay. Then again you have a cow here which is the donor cow, which has high genetic merit like the sire or the bull. And this donor of high genetic merit is made to undergo superovulation. She is made to produce 8 to 10 ova by the exogenous administration of hormones. And then you can collect the bull from this, semen from this bull, and you can conduct artificial insemination, what I have taught you few minutes before. You can conduct artificial insemination when the cow is in estrus. And you should have crossbreeds or recipients. Okay. These foster mothers, they serve as just incubators and do not make any genetic contribution towards the qualities of offsprings. So you should see to it that all these foster mothers and also the donor mother are brought to the estrus simultaneously. That is estrus synchronization of donor and the recipients. And from this superovulated female, once fertilization is over, after 6 to 8 days you can retrieve or recover the embryos. Okay. And these embryos can be put into individual foster mothers. So instead of producing one calf, simultaneously you will have 5 to 6 calves. This is what exactly is done. First part is the superovulation. Second part, when you transfer the embryos into the recipients or foster mothers, it becomes embryo transfer. So this picture nicely gives you all the steps involved in Soyet or Moyet. Same thing I have given here. So here also artificial insemination is preferred to natural mating, because ultimately the purpose of superovulation is to genetically improve the progeny and also the number. What is embryo transfer? I have explained you. Let me just brief you again. So the embryos will be developing in the superovulated cows. They are retrieved or recovered after 6 to 8 days of insemination. In cattle, recovery process is easy. It is done using a catheter. Okay. Since the reproductive system is quite big, there is no need to follow any surgical procedure. You can just suck the fetuses with a catheter and then go for embryo transfer. But on the other hand, if you are doing all this in animals with smaller reproductive tract, surgical procedures may be needed to expose the oviduct and then retrieve the embryos and then put them into the reproductive tract of the foster mothers. Okay. So then before transferring the embryos, they need to be microscopically examined. If they are fit enough, if you are confident that they will develop, only then they can be shifted to or transferred to the foster mothers. Embryos are transferred into synchronized foster mothers by a procedure comparable to artificial insemination. Then embryos can be frozen or cryopreserved again and stored for the use whenever it is needed. About 50 to 60% of the progeny can be achieved in the cattle by this method. And a superovulated female will result in 5 to 6 pregnancies. So instead of getting one calf through natural mating, through this Soyet or Moyet, you will get 5 to 6 calves. Okay. Then the resultant embryos, before shifting, based on your needs, what type of progeny you want, you can subject them to manipulations like embryo splitting, embryo biopsy and embryo sexing. Okay. Suppose if you want only males, you can collect only males and transfer the other way also, like this. What are the limitations of these techniques? Supply of embryo from superovulated donors is limited, first one. And then when you are doing all this procedure, freezing and thawing you have to do continuously. That means suddenly you will lower the temperature, suddenly you will increase the temperature. This is called freezing and thawing. So freezing and thawing of embryos requires lot of care to keep the embryos functionally intact. In this procedure, when you are doing this procedure, if the embryos die off, then whole purpose goes a waste. Then embryo transfer requires technical skill besides high cost factor. Okay, cost is also higher. These are the limitations of SOET or MOET. Dear students, I think we have discussed about 2 techniques pertaining to animal improvement. Thank you for your patient listening. Any questions you do can contact me. Till then, stay blessed. Thank you.